Okay, back to hunch a little bit more, and we're going to pick up where we left off in regards to looking at the function of the nervous system, realizing that there are a number of different component parts that we need to consider, and we'll talk about that in this short video, which we're going to do right now. First of all, I think we need to understand some anatomical terms in regards to what the nervous system is about. First of all, a nerve. The nerve is a bundle of hundreds of thousands of axons, plus associated connective tissue that's going to hold it together, as well as blood vessels. You got to remember that nerve is still living tissue, therefore living tissue still needs nutrients and oxygen and everything else that it needs to be able to survive. So there's blood vessels that will actually help to survive to supply the nerve. That's called the vasal nervorum. You don't have to remember that, but basically, so the nerve is a combination of not just the neural material, the axons or those long strands of the nerve, but also the connective tissue that holds it together, the blood vessels and stuff like that. And what happens is that nerve is held together in a bundle, which follows then a specific path and serves and will eventually end up in a certain region. And if I'm looking at a nerve, this is sort of like a diagram of what we'd see with the nerve, and we've seen this when we looked at them underneath the microscope earlier in the year in the lab. And basically what we have is we have a cell body, and this area here would be the area of the cell body with a nucleus obviously in that cell body. This may be located this will be located in various areas. A lot of them are going to be in the cerebral cortex or in the brain itself, where they'll be congregated around in a certain area. And then what happens is these little areas out here, these are all those dendrites, those little feelers that are around the outside. And what the dendrites do, it allows the communication from this particular nerve to adjacent nerves. So therefore there's a little bit of communication or a little bit of a close association between this particular nerve with all those around through these areas of dendrites, which will interdigitate or will be close to other areas. Then from that area of the cell body, again with the nucleus, which is the control center, we're going to have this long strand that's going to go, and again that strand may be short, and some neurons have very short strands, but depending upon how far that nerve has to go, there may be significant length of these, and some of these will be number of feet. In other words, if I have to go down, a nerve is going to go down to the area of the foot or something like that. What happens is once it synapses in the spinal cord and leaves the spinal cord, that nerve is just a long strand. It's going to continue all the way down to the foot where it ends up in a muscle or wherever the case may be. So that long strand that we find in here, this long strand that we find in here is called the axon. Okay, and again, multiple lengths, could be all different lengths. This axon is surrounded by a material which is called myelin, and it creates a sheath around the outside. I think the sheath is produced by certain cells called Schwann cells, and the Schwann cells make this material that make this insulating material, which is the myelin, along the nerve. Now that myelin does a couple things, and it's really sort of important for a number of different reasons. Number one, it does provide a little bit of an insulation. In other words, it does insulate the nerve so there's not a whole lot of cross communication, like just taking a bare wire. If you take two bare wires and touch them, it creates a short circuit which messes up the whole system, the electrical system. So all of a sudden this myelin will provide a little bit of insulation. But what it also does, and probably just as important if not more important, it allows nerve transmission to occur more rapidly. What they find is with this myelin, we'll find that the nerve conduction goes from one area to the other area really rapidly and jumps these areas. These are called the nodes of Ranvier, right in here and here and here and here. What they find is that when we do have this myelin sheath on a nerve, that the nerve transmission, the conduction velocity of that nerve is much faster. There are a number of diseases that people have which are called demyelinating diseases, where the myelin gets eaten away, usually by autoimmune reasons. And antibodies against their own tissue will destroy some of this myelin sheath. And what the resulting fact would occur with that is their nerve transmission is slowed, so therefore muscle function and things like that would be reduced. Okay, not quite as rapid or not quite as controlled. So anyway, that's the axon with my myelin, with my lumps of myelin and the myelin sheath produced by cells called Schwann cells, which are around the area of that axon, and then finally ends up at some end and with my nerve terminals, which come to the very end. Now these might be the area where we see attaching to in that synaptic gutter, in that neuromuscular junction or other places. And basically as an effector, or we might have these areas in an afferent nerve or a sensory nerve having areas of receptors, and the receptors then will pick up the signal and then transmit it back towards the cell body, back in the area of the brain or wherever the case may be. This is just the same other picture we have. Here's the cell body, and here the nucleus we have here. Here are the dendrites for communication with adjacent nerves. Here's the axon coming down this way, the myelin sheath that covers that axon. Those nodes of Ranvier that allow for that skipping or that jumping of that nerve conduction, and then the nerve ending, or the axon, what's called the axon terminal down here, which either can be attached in here or at receptors at the end of these axon terminal for picking up sensory information and taking it back towards the brain, or could be those areas like the end of the nerve terminal where we have that neuromuscular junction, where now we have it's the signal from the nerve actually being transmitted to the muscle. So that's what we call a nerve. Next thing we have is what's called a tract. Okay, now a tract are bundles of nerves that contain distinct bundles of axons having a common origin or destination, and therefore they carry similar information. And this is important because when we look at, let me show you a little example of what we're talking about here. Here's the spinal cord. This is a cross section of the spinal cord. Here this would be the spinal nerve coming out here. Spinal nerve would be coming out here through that area of that gap between the vertebrae, as I mentioned in previous videos. What happens is there are bundles inside that spinal cord, and these are called tracts. Okay, there's all kinds of there's multiple different types of tracts, and what these tracts do is it's sort of like a route for nerves with a common function, a common origin, or common destination to travel. It's sort of like you know, if you take the RTA down to Public Square, then you have the red line, you have the green line, you have the waterfront line, and stuff like that. It's going a specific way, and as a result, what happens? These tracts are basically like a particular line of the transportation system. They'll take nerves and bundles of nerves that come from a specific place or going to a specific destination or with a specific function to that area. And that's what a tract basically is. And that's what the brain, you'll find tracts in the brain, tracts within the spinal cord, and things like that. The third type of term we should talk about is a nucleus, and this is largely see that what we talk about in the central nervous system. Within the central nervous system, there are bundles of nerves that contain distinct bundles of axons that have a common function or destination, very similar, but located within the brain as cell bodies. Okay, so in other words, if I have a number of nerves that have a specific function, they have to start somewhere, and where they start, the cell bodies all clump together, and this area where they clump together is called a nucleus. Okay, it's called a nucleus. So it's a cluster of neuronal cell bodies within the central nervous system. If I look at a diagram of this is like a cross section of the brain. They've actually cut up out of the brain right here. This would be occipital lobe, here would be the frontal lobe, here would be what is called the temporal lobe, parietal lobe would be in here, and within the brain there are these areas of collections of cell bodies, you know, in multiple areas. You know, here, this is called the basal nuclei, which is basically a group of 3 different other nuclei which actually work together, okay, or actually are lumped together is what's called the basal nucleus. The red nucleus is down in here. These are basically clumps of cell bodies of particular axons that have a common function, okay, and that's what a nucleus is. But the nucleus is in the central nervous system, okay, it's in the brain, okay. So next thing we have to talk about is a ganglion. Now people say, oh yeah, I have a ganglion, I have one on my wrist. No, that's a different thing. That's a ganglionic system. That's a totally different thing. Throw that totally out. We're not talking about that. What happens is a ganglion is small masses of nervous tissue containing primarily cell bodies of neurons that are outside the brain and the spinal cord, where the nucleus was inside the central nervous system. A ganglion is outside. Now this is a diagram of what we see. Here's the spinal cord again. Here's nerves either entering or leaving. What happens, this is called the dorsal root ganglion right here, and this is where cell bodies are. So what's going to happen is we'll see that there's what's called a synapse. A synapse is a word for a connection where one nerve actually influences another. So here's an area where the synapse is, and there's a cell body that would be outside. If we open up and looked at the nerve as it leaves the as it leaves the spinal cord, you'll see a little lump on the back side, the posterior side. So this is posterior here, anterior would be out in here. And what happens is this area the cell bodies are clumped together and creates a little knob on that nerve, and that's called a ganglia. So a couple of things about neural connectors. Again, when we talked about the neuron is just another name for a nerve. So we talked about what that structure of the nerve. Neurons could be multipolar, they could be bipolar or unipolar. The diagrams that we showed before were basically unipolar nerves. In other words, they had a cell body, an axon, and then dendrites from the cell body, but then at the end they have the nerve terminals or the nerve endings on one end. So there's one axon coming from one cell body. However, I could have a bipolar nerve where I could have a cell body, and I could have an individual cell body, and then like here, and then I have an axon going one direction and an axon going another direction. It's because it's bipolar. Bi means two. We know means two. So it goes in two different directions. Or it could be multipolar with actually multiple chains coming off that cell body. So neurons may be, many of them are unipolar, but they also may be bipolar or they be multipolar. We also have what are called interneurons. Okay, and interneurons there's a lot of these, and what they are is they're short connecting neurons. They're short connecting neurons. Right now, while I'm home trying to amuse myself in other ways other than making videos for A&P and midterm, I'm actually going to put a new stereo in my car. And what happens is these interneurons are basically taking a little jumper wires. I'm connecting one wire to another in these little jumper wires, and that's what these interneurons are. Basically, they're shorter neural connectings. It's a short way to connect one neuron to another neuron with a very short interneuron between that. Another neuron I should mention are called neuroglia. Initially, the thought about neuroglia has changed over the years. Glia actually means glue, okay? And a lot of times they thought the neuroglia were basically sort of cells in the brain and in the neurological system that basically held things together. That really did not much else than that. That they thought they really didn't generate or propagate any action potentials. Again, when I talk about action potential, I'm talking about that electrical signal that's going along the nerve. However, I think that we're hearing a lot more evidence now that they do a lot more than that. They're able to transmit signals, and they do have some transmission function, which a lot of times people in the past thought didn't happen with neuroglia. They're able to multiply and fill spaces formerly occupied by other neurons and things like that. There's a particularly virulent form of a brain cancer, which is called a neuroglioma, which is a highly malignant brain cancer. Life survival, the life expectancy after the diagnosis of neuroglioma or glioblastoma is probably only about a year and a half. I had a cousin who actually passed away from a glial type tumor in his brain, and the problem is because it's not confined like in a nucleus where we have one confined area, they're all over the place. It's very hard and difficult to control it. Those are called neuroglia. So we have neurons like we've talked about before, interneurons, which are very short neurons that are connecting neurons, and neuroglia, which still there's a little question about what their overall function might be to give it all the credit they're due. And again, this is just looking at, let me get rid of these marks that I made before. Whoops. Do that up. There we go. We're getting rid of those. Again, this is a unipolar neuron, again with the cell body up in here and the dendrites, the axon, the nerve terminals coming down here. This is just to look at that axon, that myelin sheath with the Schwann cells and nodes around here. And this is basically that bipolar neuron. We have an axon that goes one way and an axon goes another way. That's all that shows. Okay. We should also mention that nerves are especially, we're talking about the afferent nerves, A-F-F-E-R-E-N-T nerves, and basically these are the ones that are picking up signals from various areas of the bodies, and they're picking up signals by the ends which are called receptors. Okay, now we have three types of receptors: exteroreceptors, interoreceptors, and proprioceptors, and we'll talk about those. Exteroreceptors are basically receptors that are picking up stimuli or sensations near the surface of the body, so they detect from the outside the body, the surface of the body, or our external environment, such as smell, taste, touch, pressure, vibration, temperature, pain. All these things are things that are around us, and basically these exteroreceptors will pick up this and will send an afferent signal from the outside or the surface of the body back to the brain to give it information, again going back to our interest in what we call homeostasis. Interoreceptors are internal, okay, and these interoreceptors are like in the walls of blood vessels, in the walls of my intestine, in the walls, or all over in the nervous system. They're sampling internal activity, pressure, chemical nature, constitution, constituents, things like that, to be able to provide then an afferent signal back to the brain if I need to make a change. Typical example, if let's say there's an area that needs more blood supply, what we're able to do is if there's probably the best indication is like in cold, and we'll talk about this in just a second. If we're cold, we know that what happens is the blood vessels around those areas that are cold will constrict. They actually narrow down. Well, what really does that? Actually, that's the cold will cause a stimulus, and inside the blood vessel, then that would be an exteroreceptor. But the blood vessels then have receptors that will actually detect certain levels of what they need. So in other words, if I need more, if you look at cold, you'll find that pallor, and that's the exteroreceptors. However, down the road, the body says, hey, this area is cold, and and we have to rewarm it, because if it's too cold, it needs to be rewarmed. And you notice when your hands get cold, all of a sudden when they start to rewarm, it turns bright red. Well, these interoreceptors are saying, let's increase the blood flow here. They're detecting that they need more blood flow because the tissues are saying, hey, feed me, Seymour, I need the oxygen. As a result, what happens is it sends a signal to the brain, the brain sends a signal back down saying, okay, blood vessels dilate. So basically these interoreceptors are basically in blood vessels and viscera and other places of the body, and they're detecting internal stimuli, sending that afferent signal to the brain. They're generally not felt. We don't understand those. If you have a stethoscope and you place it over your abdomen, it might be really silent. You might not hear anything, but if I take that stethoscope and actually press down on my abdomen and just push a little bit, then let go, what you actually hear is you'll start to hear a gurgle squirting around, and basically you're stimulating some of those nerves in the wall of the intestine saying, hey, let's move things around because there's been a little pressure. So these are inside interoreceptors, proprioceptors, okay, and proprioceptors are really something that I've had a lot of activity with. Proprioceptors are basically sensory receptors that are located in muscles, tendons, and joints, as well as in the ear, but I'm not going to go to that. We'll talk about the muscles, tendons, and joints. If you've noticed that when you're walking along and the sidewalk's a little curved or a little crooked, and what happens, you step, and all of a sudden your foot goes in underneath it, but what you'll realize is the foot then all of a sudden, without you thinking a whole lot, tries to go back underneath you to keep your foot straight up and down so it doesn't continue to turn in. It's that rapid muscular response. Well, the reason why that's like that is these proprioceptors are able to sample position of the body in space and joint position. And as a result, what happens is when that position, when those proprioceptors become stretched or stimulated, it causes a signal, an afferent signal that goes to the brain and says, hey, we better move that foot back under where it's supposed to be so it doesn't continue to turn in. And then all of a sudden, without me thinking about it, the foot's going to turn in. And basically that's what we find. I used to have a lot of kids who would be sent to me or come to me because what happens is they had an ankle sprain and they had been to a doc, and the doc says, well, just don't use it for a while, it'll get better, all ankle sprains get better, and just give it some rest. They don't give them any rehab program. And what happens is we find out that their proprioceptors have been damaged, and what we do is we have them stand. If you could try this. If you stand up, okay, and lift one leg up and stand on one foot, basically you'll see you'll actually feel these little small twitches of the muscle as you're trying to balance yourself. Well, what happens is your balance is improved by a couple things. Number one, your ears, okay, which is helping, and that allows a little bit of a balance, as well as your eyes. Your eyes are sighting the horizon, so by sighting the horizon, you're able to help and improve your balance. But the third thing that's important are these proprioceptive fibers in the ligaments and the tendons and everything around your ankle. And as a result, what happens is after injury, these get torn or ripped. So what I used to do is have people stand on one limb to build the strength of the muscles back to where it should be, so they could do that with their eyes open initially and with their and obviously with their ears. But then what happens is that after they could stand for a certain length of time with their on one leg with their eyes open, I have them do it with their eyes closed. Now, if you try that with your eyes open, your eyes closed, you'll see how much more difficult it is with your eyes closed, because you're relying not on your visual stimulation to keep you balanced, but now only on those proprioceptors. Also, I mentioned earlier in muscle's always under a little bit of tension. We looked at those sliding filaments. There's always a little bit of an overlap. Okay, what happens is that's because the muscle is doing constantly contracting and relaxing to just a little bit to create what we call tone. The muscle tone is actually produced by two things. One's called a Golgi tendon, and the other one's called a muscle spindle. One contracts, one relaxes, and measures tone, and they battle each other to try to keep the muscle so it's mildly under a little bit of tension where it should be. Okay, and that's what we call muscle tone. And these proprioceptors are very important in helping to manage that. So these proprioceptors are really important. People don't talk about it very much, but they're really important in sampling body position, tension of muscle and things like that, joint motion, as well as helping in my equilibrium. All these things are important in regards to looking at proprioceptors. So exterior receptors on the external surface of the body, sampling my environment. Interior receptors, sampling things on the inside, blood vessels, intestines, all kinds of stuff like that, as well as proprioceptors in muscles and joints, and helping to adjust the body's position in space. Okay, a couple sensory receptors we talk about, free nerve endings. And free nerve endings are basically bare dendrites. When we get to the end of a nerve, what will happen is some nerves just end in just fibers. Things such as pain, temperature, a tickle, itch, and some touch basically have bare dendrites near the surface of the skin that are able to detect these stimuli to send an afferent signal to the brain. Okay, actually it turns out that things like tickle and itch are basically lower levels of pain. Okay, when we look at pain, actually the tickle and itch are very closely related to pain, just to a lesser degree. We also have what are called encapsulated nerve endings, and encapsulated nerve endings are basically dendrites, but they're closed in a capsule, and that capsule is able to respond to things like pressure. So therefore, encapsulated nerve endings are very important in something such as touch. We also have separate cells, and some of the receptors that we have that will actually send signals to the brain, afferent signals to the brain, really aren't so much part of an individual nerve itself, but actually receptors. And what happens, these receptors are next to and have and synapse, and again synapse means sends a signal to or associates with what are called first order neurons, or neurons that come right from there. And basically that's like in the retina. When the light comes in through the pupils and it gets focused by the lens to the back of the eye, to the retina, what happens is certain cells, we talk about rods and cones and stuff like that, these cells actually create a chemical response that then synapse or send a signal to the nerve, and the nerve then goes through those optic canals, optic foramen, to back to the area occipital lobe to be able to see. So basically, the photoreceptors basically are specialized cells that will synapse with a first order neuron, or the first neuron that goes back afferently back towards the brain. Same thing happens with the inner ear. They're called hair cells. And these hair cells, the inner ear looks like a little conch shell. It has little circles in it, and these circles, this tubular circular area of the inner ear is filled with fluid, and there's like a band inside there. And from the band, there's small little fingers that stick up, little hair cells. And on top of the hair cells is like a little knob. What happens is that little knob is going to move. When sound hits my eardrum, causes my eardrum to vibrate, which then causes a transmission of vibration of all the small bones, the three small bones in the middle ear, that takes it to another membrane, which is at the beginning of the internal or the inner ear, and that causes that membrane to vibrate. When that membrane vibrates, it sets up a fluid wave signal through this area called the cochlea, and that cochlea then causes a fluid wave around there, which causes these little inner ear hair cells to bend because of this little filaments on on the top, and as a result, that's how I hear. Also, taste buds. Taste buds are separate cells that detect chemicals in our mouth. It's sort of interesting when they talk about how things taste. If you get a burger at McDonald's, it tastes different from burger at Burger King, okay? And other places, Five Guys, Brown Bag, whatever the case may be, they all taste a little different. Why? Because what happens is there's a whole science in regards to creating chemicals that improve taste. In other words, if you look at a lot of the food you eat, they'll say artificial flavorers or stuff like that, artificial flavoring. And basically, they create chemicals. And because these chemicals, when they get on the tongue, they stimulate these taste buds, these gustatory receptor cells that then cause a change, sends a signal to various nerves that send an afferent signal to the brain, and that's where I get taste. Okay. A couple types of stimuli that we have: mechanoreceptors. Mechanoreceptors are receptors that pick up mechanical pressure, touch, pressure, vibration, proprioception, again, that position, sense, hearing, equilibrium, stretch of blood vessels, internal organs. Something that's mechanical are picked up by receptors called mechanoreceptors. Obviously, thermoreceptors, temperature, because thermo means temperature. Nociceptors. Nociceptors detect physical or chemical damage to tissues. When we have damage to tissue, inflammation occurs. And when the inflammation occurs, various chemicals are produced in those tissues. One of the things we talk about are prostaglandins. Prostaglandins are basically pain-producing type chemicals. And these prostaglandins that are produced are picked up by the nociceptors, and that's why things that are inflamed are painful. Why? Because there's a vasodilation, the vessels dilate a little bit, but what happens is all these chemicals that are there, bradykinin, prostaglandins, all these things are basically creating a chemical change in these nociceptors. The chemicals are there cause a change or stimulation of these nociceptors, and that's what causes the pain. Photoreceptors, basically these are the receptors that we see like the rods and the cones that are affected by light when it hits the retina. Chemoreceptors, again, chemicals in the mouth like we talked about with taste, also with the nose with smell, and also various body fluids are because the composition of the body fluid is made up by certain things. So as a result, that's where these chemoreceptors come into play. Osmoreceptors are involved with osmotic pressure, and we'll talk a little bit more about that when we talk about the cardiovascular system down the road. Let me just mention how this all comes together. Thermoreceptors basically end up in the hypothalamus, okay, but they originate as thermoreceptors in the skin, okay, and what happens is the control areas for my body temperature are in what's called the preoptic area, which is in front of the optic region. There's a heat promoting center and neurosecretory cells in the hypothalamus and the pituitary gland that help me to control my body temperature. The body needs to have its core temperature, the center portion of the body, stable. It needs to be consistent, and the reason why it needs to be consistent is the body likes to function at a particular temperature. All the body chemicals and body reactions occur better when they're at a particular temperature. We don't want to vary that temperature, so we try to keep our core temperature, the center of the body, consistent. If I'm in a cold environment, what happens is the body says, hey, listen, we got to keep that core temperature consistent, and if I'm exposed to this cold, that core is going to be cold, and therefore my body processes are going to go down, and as a result, that might be detrimental. So what it does is it shunts blood from the extremities, decreases the blood flow to the extremities to be able to take more blood, which is warmer, to the area of the core, okay? So as a result, what happens is when I'm outside in the cold and getting cold, the effectors, okay, basically cause a vasoconstriction in the skin to decrease heat loss. Again, the receptors picked up this lower temperature, sends that afferent signal to the brain, then the brain sends out afferent signal to effectors that causes the vessels in the extremities to vasoconstrict, to get narrow. Let's move the blood from the arms and the legs and the ears and the nose and stuff like that and put it in the core, in the center portion of the body. The adrenal medulla also releases various hormones. We talked about how that neurological system is associated with the endocrine system. The adrenal medulla is part of the endocrine system, and what happens is when I'm cold, the adrenal medulla, under the stimulus of the neurological system, then will secrete things like epinephrine, norepinephrine, which then increases the metabolic rate. So these hormones will increase level of metabolism, as well as the thyroid gland down at the bottom. The thyroid gland secretes thyroid hormone, and what the thyroid does is the thyroid gland controls metabolism. So therefore, if I create more thyroid, or if I secrete more thyroid hormone from the thyroid gland, my metabolism rate goes up. What does metabolism do? It works, therefore that work produces heat. Okay, and then we also know that the skeletal muscles will actually be forced by effectors to contract. And what they do is they'll start to rapidly, rhythmically contract, which we call shivering, and that shivering is basically another way to produce energy or produce heat. Okay, so we actually make heat by that muscle contraction, that muscles working fast, and therefore the shivering will also generate heat. So that's how the neurological system works, again, in this case also with the endocrine system down here with the thyroid gland and the adrenal medulla, to try to keep the body in homeostatic control, which is what living is all about. So in summary, what we've talked about today, we've talked about the different parts. We've talked about nerves, we've talked about tracts, we've talked about nuclei, we talked about the ganglion, we talked about how the body controls itself by various receptors, samples external environment, samples internal environment, and constantly every minute or every second of every minute of every hour of every day sending signals, afferent signals to my brain with all this information. My brain then integrates that, puts that all together, decides what's right, what's wrong, and stuff like that. If it's right, hey, if it's good, leave it alone. But if it's bad, what to do? And then sends out an efferent signal from the brain to wherever it needs to do to try to make a change or alter some change to keep that homeostasis under control. And this is pretty much what we talked about today, and hopefully this has been helpful in understanding all that. We'll talk to you a little bit later about a little bit more of the divisions of the nervous system, such as the somatic versus the autonomic nervous system. We'll talk about that a little bit later.